many things about coronavirus comprehensively. The second theme is a bit more specific. It is about vaccines. The second presenter will be Director General Dr. Jerome Kim of International Vaccine Institute. And it is a leading international organization located in Korea. And he's now Director General of IVI and Uniformed Services and University of Health Science. And he's an adjunct professor. And also, he is an adjunct professor in Seoul National University. He earned degrees in Hawaii University in medicine and history, and also he earned doctor of medicine degrees in the United States, and also he has many international careers as well. The theme of his presentation is COVID-19, the global pandemic and progress in vaccine development.
I'd like to thank the organizers of the Big C for the opportunity to speak today virtually on the topic of COVID-19, the global pandemic, and progress in vaccine development. Every day we see a worsening of the COVID-19 pandemic, with total infections approach in excess of 20 million people and deaths in excess of 700,000 people. We daily see the impact of failed policy to control, to suppress the COVID-19 transmission around the world. First, a bit about coronaviruses. Coronaviruses are very unusual. They are what we call promiscuous in that they can affect multiple different members of different species of animals. So, for instance, those of you who remember the SARS outbreak in 2002 know that SARS originated in bats, transitioned to palm civets, which are a type of ferret, and then transitioned from palm civets to human beings. MERS, on the other hand, again, started in bats, transitioned through camels to infect human beings. And Korea was, of course, affected very gravely by the largest outbreak of MERS outside of the Middle East. SARS-CoV-2 presumably also originates in bats through some unknown intermediate host, perhaps spiny anteaters or pangolins, and then to humans. Humans are very efficient spreaders of SARS-CoV-2. They're not terribly efficient spreaders of SARS or of MERS. SARS-CoV-1, the original SARS outbreak, and SARS-CoV-2 share a genetic similarity of roughly 80% at the sequence level, and particularly in the pink colored spike proteins shown in the bottom left-hand part of the screen. The spike proteins share roughly 70% homology in what we call the S1 gene. There are to date 6 known circulating strains of SARS-CoV-2. We don't want to call them species or even, as we often do in HIV clades, because the amount of genetic difference between the different strains is actually pretty minimal, on the order of 6 to 7 individual nucleotide changes in a genome of roughly 30,000 base pairs. So this is actually a very, very small amount of variation. And the other important thing to remember is that the mutation rate for SARS-CoV-2 is 10-fold less frequent than is seen in influenza. You can see that a bit better on this illustration. On the far left, you see the amount of genetic variation, a genetic tree for SARS-CoV-2. That's the little thing the size of the head of a pin. What you see in the middle is a genetic tree for a single year's worth of influenza virus. And so the bigger the tree, the more genetic variation there is. And you can see, the reference line at the bottom, 0.1, reflects a standard measure of genetic distance. And you can see what it is then in 1996 for the hemagglutinin protein of influenza. What you see on the right is the genetic tree in 1996 for HIV-1 sequences from the Democratic Republic of Congo, so a single country. And you can see the huge amount of genetic variation that's present in HIV compared to influenza, and the great amount of variation there is in influenza compared to SARS-CoV-2. So although we say SARS-CoV-2 doesn't seem to mutate much, that there isn't much sequence variety, we still don't know whether that will mean that when we develop a vaccine, will that vaccine be like rotavirus vaccine, which hasn't had to be changed in over 13 years and is still highly effective in controlling rotavirus diarrhea? Or will it be like influenza, where we have to switch out individual strains of SARS-CoV-2 or influenza every year? This shows the epidemic curve for Korea, and I think everyone remembers what it was like in Korea at the end of February and the beginning of March in 2020, where daily the number of cases were increasing. And then, of course, the actions of the Korean government and the Korean people allowed SARS-CoV-2 to be brought under control until we got to April, where the number of infections was actually very low, approaching zero, and actually zero on several days in April. And then as the government released some of the restrictions on activity, the number of cases increased, but the Korean government had set a target that the number of new cases should be less than 50, and that the number and the percentage of cases for which no known attribution of infection could be found was less than 5%. And since then, Korea has been in a state of a very low continuous level of infections. Occasional outbreaks, the Itaewon outbreak, an outbreak in an e-commerce hub, outbreaks in churches, but really the government has been able to keep the number of new infections under 50 a day. And that's even more true if you look at the number of cases that actually originated in Korea, what we call local infections. A lot of the infections in Korea are coming from people who are infected in whatever their home country is or wherever they were visiting and come back to Korea and have to be quarantined. So a lot of the cases, of the new cases, actually over 50% of them recently, have been due to cases that are not originating in the Republic of Korea. So again, a very good job of maintaining a very low level of infections. And I think, as many countries are experiencing around the world, the release of restrictions, release from lockdown, has resulted in a huge surge of infections, for instance, in Spain or in the United States. And this has dramatic implications for the health of the people and for the number of deaths that are being reported around the world from COVID-19. Korea has, on the other hand, been able to maintain a very low and steady amount of infections, and hopefully it will remain that way until a vaccine can be found. So, Koreans are able to go to church. They're able to vote. They're able to take tests. They're even able to play baseball, things that other countries around the world really have not had the benefit of. And I think the other thing that we remind people about when they ask us about Korea is that Korea never experienced a true lockdown. Although the government had put into place a number of different restrictions, there was no lockdown. Koreans were still able to go to restaurants, to go to the supermarket. There were, except for a very brief period, you were able to get masks and hand sanitizer, things that in many places of the world remain in short supply. So the South Korean model of control begins with preparation. And we often forget to emphasize the importance of the prior experience with MERS in 2015. As a result of that, and a result of criticisms of the government response to MERS, the government made changes in the Epidemic Preparedness Law, the Infectious Diseases Prevention and Control Act of 2010, that allowed the government then to have much better ability to trace people, allowed the government to practice what they call tabletop exercises. So in December of 2019, the government undertook a tabletop exercise with leading academics around the country of a Korean couple who returned from China with an unknown pneumonia. Little did they know that a month later, the world would experience the first outbreak of SARS-CoV-2 or COVID-19. So another important element of the Korean approach to COVID-19 was having clear command, control, and communications. So command starts at the Prime Minister level. It's been delegated to the head of the Korea CDC, the Vice Minister for Health. And that approach of having governmental control from the top all the way down to the areas where government policy is being implemented has been a model that has been successful not only in Korea, but in other countries in the world where that approach was used: Australia, New Zealand, Iceland, Taiwan, the Republic of China, the People's Republic of China. Again, emphasizing that democracies have been able to accomplish this if they've been properly organized, communicable disease prevention efforts are properly communicated consistently to the population. The other part of this, of course, is what the government calls test, trace, and treat. Not to forget that they also did a fair amount of isolation. So those people who are identified as COVID-19 positive were isolated for 14 days, sometimes longer, until they had 2 negative COVID-19 RT-PCR tests 24 hours apart. So again, the Korean government put into place a very effective strategy for testing, tracing, treating, and also the process was transparent. Every day, the Ministry of Health and Welfare briefs the Korean people and anyone who happens to tune in on the English language channel, Arirang, about the ongoing state of the COVID-19 pandemic and what the government is thinking and doing to deal with new contingencies. They continue to emphasize distancing, masks, hand and cough hygiene, and all of this could not be possible without the intense cooperation of the Korean people. So again, society needs to get around this and support the government activities. In countries in the world where the social policy and the government political policy is failing, there isn't often the same level of coordination that you saw in Korea. This describes some of the efforts that the Korean government has done since releasing a lot of the social restrictions. And is often the case, there was a lot of planning that went into deciding when to release the restrictions, what types of restrictions should be put into place, and what the different levels of restrictions are. So that you can see that at level 3, which is where we are now, gatherings of up to 100 people are allowed. You are allowed to have limited attendance at sporting events and other activities. As we get more severe, so if there were a significant outbreak in Korea and the pandemic were no longer under control, the Korean government would ratchet up the level of social restriction until we would return to a situation very similar to what we had in April and March, where there were a lot more restrictions in place on gatherings and working and sporting events and school. So again, success in controlling the outbreak, not eliminating it, but keeping it under 50 new infections a day with less than 5% of cases untraceable, has allowed the government to release a lot of the social restrictions, allowed children to go back to school. So now we are going to switch a bit to COVID-19 vaccines. And what you see in the upper left-hand corner is the Brooklyn Bridge during the age of COVID, empty, no people. How are we going to get back to the Brooklyn Bridge as it usually is on a busy weekend, crowded with people? Well, is a coronavirus vaccine itself the bridge to what we call the old normal? One of the first things that you need to do when you look at a new vaccine is to think about what are the key questions. Probably the first question that we ask is, does infection, in this case with COVID-19, provide immunity against reinfection? Now, the answer to that is we believe that it is so. But why is it important to know? Well, in a classic vaccine disease model, when you take the disease, and let's take hepatitis A or polio, different people respond to the virus in different ways. A person with hepatitis A may be relatively asymptomatic or may progress to full jaundice and liver dysfunction, and in some very, very unusual cases, occasionally die of hepatitis A. But in almost all cases, when the person survives, the body's immune system is able to control the virus, suppress the virus, and eventually eliminate the virus. And those same immune responses, those protective responses, protect the body against reinfection. So a person who's acquired hepatitis A infection naturally will not be infectable by hepatitis A again. The same is true of measles. The same is true of rubella. The same is true of polio. So again, infection provides immunity against disease. Why is that important? It's important because vaccines often mimic disease. It gives you the benefit of disease without actually getting sick. So in all those models where we've been able to successfully develop a vaccine relatively quickly, infection provides resistance to reinfection. Is that the case in COVID? We believe it's so. And for that reason, we think that it's feasible that a COVID-19 vaccine could be developed relatively quickly. So what are the data? There have to date been 3 monkey challenge studies. So monkeys are a species, are a group of mammals that can be infected with COVID-19 and develop a mild pulmonary disease, much like the asymptomatic humans who acquire COVID-19 infection. In those 3 studies, the monkeys were challenged with COVID-19. They were infected. They were allowed to recover, and then they were re-challenged with virus. And in those cases, the monkeys were protected against reinfection, all 3 studies. So now we have 3 studies in monkeys. Another group of animals that can be infected by COVID and actually develop fairly severe disease and can actually sometimes die are hamsters. So Syrian golden hamsters can be infected by COVID-19. If you infect a hamster, it survives. That hamster is subsequently resistant to future infection, so to re-challenge. That again is another animal model that would suggest that, like humans, hopefully, once you acquire COVID-19 infection and you recover from it, you're resistant to reinfection. That would imply that a vaccine may be relatively easier to develop. A second question that's come up that's kind of related to this is, well, how long will that protection last? And you know, with some viruses, protection against reinfection lasts a lifetime. So once you've been infected, you're protected with measles, for instance, you're protected against measles for the rest of your life. That is not always the case for some of the common cold viruses or some of the coronaviruses that we think affect people on a seasonal basis. They've done one study in monkeys where they vaccinated the monkeys with a DNA vaccine, and they waited 12 weeks and then re-challenged the monkeys, and those monkeys were protected against reinfection. That raised the possibility that although there weren't a lot of visible antibodies or visible T-cell responses at the time of re-challenge, that immunological memory was able to control infection upon exposure to COVID-19. We hope that's the case for humans as well. The second key question is, which of those protective immune responses are important? Typically, we say it's antibody. So neutralizing antibody in particular, antibody that binds to and neutralizes a virus. But it could be other things as well. In fact, there's a study using a DNA vaccine that would suggest that not only neutralizing antibody is important, but some of the other functional antibodies, antibodies that mediate things called antibody-dependent complement deposition or antibody-dependent cellular phagocytosis. Those things were also correlated with protection. They are not neutralizing antibodies, they're non-neutralizing antibodies that use the functional end, the Fc portion of the antibody molecule. And that's shown in the figure at the bottom. We think that cellular immunity may be important as well. It usually is in order to generate strong immunological memory. Killer or cytotoxic T cells may also be important, but again, we don't know the true value of those things as a correlate of protection. Importantly, it may be that a balance between type 1 and type 2 T helper cell responses may be critical in preventing a particularly severe form of enhanced respiratory disease after COVID-19 challenge and vaccination. Another important question, something that we immediately look for as soon as we're looking for a new vaccine is there a reliable animal model? And Professor David Weiner has once said that mice lie, monkeys exaggerate, and only humans tell the truth, meaning that ultimately, no matter what we see in hamsters or in non-human primates, we're going to have to test the vaccine and prove that the vaccine is safe and effective in humans. We know that you can infect a lot of different species with COVID-19. So unlike HIV, where there is no real animal model for HIV itself, you can infect monkeys, ferrets, hamsters, mice that are transgenic for the ACE2 human receptor, and you can even adapt SARS-CoV-2 to efficiently infect regular mice, now without ever having to do a transgenic mouse. Whether any of these models actually will tell you that a vaccine will protect humans, we don't know. And in fact, we need to depend on the human model. We have to have a vaccine that prevents infection in humans and then go back and establish the same model, validate the model in animals. So do we have any suggestive information? Yes, in fact, there are now 7 studies in monkeys that show that vaccines of different kinds, so you can see them listed down there, whole inactivated vaccine, chimpanzee adenovirus vaccines against COVID-19, RNA vaccines against COVID-19, Ad26 vaccines against COVID-19, DNA vaccines against COVID-19, all of them, when used in monkeys, protect those monkeys against COVID-19 challenge, which raises the possibility, again, hopeful, that within 12 months or so, so I guess by the end of this year or beginning of next year, we might be able to prove that a vaccine prevents infection or disease and is safe. Are there safety issues? Safety is a major concern, and what you see on the left is a picture from the 19th century, the 1800s, when they were first beginning to use the cowpox vaccine against smallpox. So against the smallpox disease, it was prevented by a related virus called cowpox. And when it was first introduced, people were very concerned that cowpox would cause the eruption of little cows on their arms. This was an early anti-vaxxer movement, and you can see in the cartoon that as people are being vaccinated, they're looking at their arms and their cows' heads springing out of them. Now, that was a fear. It was unfounded. There was no basis in reality for that, and in fact, smallpox has been eliminated, has been wiped off the face of the earth by that very same cowpox vaccine. So again, we have to remember that if we base vaccination and vaccination programs on strong science, on safety and efficacy, we will be able to bring COVID-19 under control when we have a vaccine. But there are two potential hypothetical safety concerns. The first is something called antibody-dependent enhancement, and it's been a concern. It's been seen or reported in dengue, it's been feared for HIV. In fact, we haven't seen real evidence of ADE, antibody-dependent enhancement, in HIV. There may be a case of antibody-dependent enhancement seen with the Sanofi dengue vaccine. In particular, as the titers of neutralizing antibodies wane over the course of 2 to 3 years, people who have not been exposed to dengue before and have just received the vaccine appear to have a more severe form of infection. And for this reason, when we look at SARS-CoV-2, we look back to the original SARS virus and to MERS virus, and in both those viruses, laboratories around the world had reported potentially the presence of ADE, antibody-dependent enhancement. Now, it may be less of a concern because after they found that phenomenon in the test tube, they were able to vaccinate animals and then challenge them with virus. And in fact, they saw protection against infection. So antibody-dependent enhancement, if it occurs, doesn't occur immediately after vaccination. Whether it occurs later is not known. The second and potentially more troubling thing was that in certain animal species, particularly in mice, and particularly with the original SARS-CoV, the 2002 SARS, if you vaccinated a mouse and then challenged them, they wouldn't get infected, but they would develop something called enhanced respiratory disease. And that enhanced respiratory disease resulted in the influx of eosinophils into the lungs of these animals. It was also seen, but less consistently, with ferrets and in monkey studies, with SARS-CoV-1 in particular, with MERS less frequently, and in SARS-CoV-2 and COVID-19, in those 7 monkey studies that have been reported, the investigators looked for evidence of ADE, antibody-dependent enhancement, and also of enhanced respiratory disease, and they found no evidence of it in the 7 monkey studies that were done for COVID-19. So it may be less of an issue, but it still means that we will have to be absolutely sure that when we vaccinate a volunteer in a study with a new COVID-19 vaccine, we commit ourselves to following them up for an extended period of time to ensure that nothing bad happens, that we don't see ERD in any of these volunteers. So we're often asked now, well, okay, I understand all the questions. How long will it take to get to a vaccine? Under normal circumstances, 5 to 10 years. But of course, these are not normal circumstances. The cost of developing a vaccine, $500 million to $1.5 billion, and the failure rate going from the laboratory to licensure of the vaccine is 93%. So only 1 in 10 vaccines that start off in a laboratory in a vaccine company actually make it and are sold as vaccines on the open market. Vaccines start in the discovery phase in a laboratory where a vaccine is created at a lab bench. That vaccine is then transitioned to preclinical studies, that is testing in animals, and then into the first of 3 phases of testing in humans. In phase 1, we look at safety. So in usually less than 50 people, you look to see whether the vaccine does anything bad in 50 volunteers. We can also get a little bit of information about how the vaccine works, what immune responses, antibody or cytotoxic T cells are generated, but really we're focused on safety. In phase 2, we look at safety and protective immune responses, humoral neutralizing antibody, cytotoxic T cells or helper T cells in the target population. typically in several hundred volunteers, 400 to 600 volunteers. And then if all those responses are adequate and the vaccine appears to be safe, we move into phase 3. Phase 3 is a test of efficacy and safety. That is, does the vaccine protect against disease and infection, and is the vaccine safe? Usually in thousands of volunteers, sometimes tens of thousands of volunteers. And you can see why then this sequential approach can take a long time. As you're waiting for one phase to end and the next phase to begin, a lot of work will be done, and that's why the cost ends up being so great. And because the cost is great, companies are very reluctant to take forward vaccines that they aren't sure about. So they really want to make sure that they optimize everything at each step before moving on. Now, we don't really have the luxury of time in COVID-19, and what people are proposing is that within 6 to 18 months, we'll be able to do what normally takes 5 to 10 years. And how would that be done? So this is a hypothetical cartoon of a 5-year development cycle for a vaccine. So you can see we do the preclinical studies. Phase 1 starts in year 1. We analyze safety. Once we're done with that analysis, and if we're satisfied, we move on to phase 2, which also takes a year. We analyze the safety and the immune response data from that phase 2 study. If it's suitable, then we move on to phase 3. Phase 3 takes 18 months to 2 years. If we demonstrate safety and efficacy, then we move on and register the vaccine. We apply for a license to do it. Now, we don't have that kind of time, and how are we going to shorten this into 12 to 18 months? Well, we overlap the phases. So you can see phase 1 starts once we're convinced the vaccine is safe. So 1 month after the final dose is given in phase 1, if it's safe, we move on and start enrolling phase 2. Within a month after the final dose is given into the last volunteer in phase 2, we can analyze safety and immunogenicity, and if everything looks good, then we move on to phase 3, which looks at efficacy and safety. So as phase 1 is going on, we start phase 2. Before phase 2 is done, we start phase 3, and hopefully we will see an efficacy signal by looking at the first available time point in phase 3, which is often a few months into the study, and I think people are hoping that we will see the vaccine against COVID-19 is working at that early time point. Hopefully then the vaccine will be moved on to licensure. So we can shorten the clinical trials period. We're looking at the same endpoint, efficacy. Does the vaccine prevent infection or disease? But we won't have the same period of time to look at safety questions. Normally we would have 5 to 10 years to establish the safety of a vaccine. Now we have 12 months. So we're really going to need to commit to longer follow-up of volunteers in phase 1, phase 2, and phase 3 efficacy studies for any of the COVID-19 vaccines. So right now there are over 200 potential vaccine candidates for COVID-19. And I'm not going to go through them all. In fact, the only way that I can keep track of them is to look in Wikipedia, which actually has a weekly updated list of all the different types of vaccines that are moving forward. And you can see the list from August 6th. Describing all the different vaccines that are in various stages of clinical development. And then the 25 that are currently in human clinical testing are shown at the right. And again, I'm not going to go through them all, but you can see them here on the slides. And how are you going to fund all this work? Well, it's a huge amount of money. The US government has committed $8 billion to develop COVID-19 vaccines very quickly. CEPI has put in $1.4 billion. You can see the breakdown by company. AstraZeneca got $1.2 billion. Novavax received $1.6 billion. Sanofi, $2 billion. Emergent, Ology, and SIO2 are getting $782 million in order to do contract manufacturing for successful vaccines. So essentially, the US government is accelerating the technological solution for COVID-19, a COVID-19 vaccine, by de-risking the process, by giving companies huge amounts of funding upfront in order to de-risk the process of development, to encourage them to get to the answer quickly, to get to the point of safety and efficacy as quickly as possible. So to accelerate, de-risk, and how do we de-risk? We provide funding to the companies. This is a different way of looking at the pipeline. You can see a few of the vaccine companies, Moderna, for instance. The first circle is when they first started their first-in-human trials. The second circle is when data first became available. And the hatched bars are when the companies intend to start production. So Moderna intends to start production of its vaccine before it even knows that the vaccine is safe and effective. And how is it going to do that? Well, the US government is paying for it. The US government has said that it will provide it will actually start the manufacture of hundreds of millions of doses before the end of this year in order to have vaccine ready when and if the vaccines are shown to be safe and effective. And what you see at the bottom is some of the other companies that are also involved in the search for a COVID-19 vaccine. So this gets into some of the data, and I don't have time to go through all of it in great detail. These are two studies of the 7 or so from non-human primates, one using the Sinovac vaccine adjuvanted in alum, so it's a whole inactivated vaccine. The second uses the Oxford chimpanzee adenovirus vaccine. And what you can see, and I'm going to circle or point out the key feature here is, you vaccinate the monkeys with either the Sinovac vaccine or the AstraZeneca vaccine. They generate neutralizing antibody. We know they generate cellular immune responses. We also know that they're protected against infection, severe infection in the lung, in fact. But the big difference is if you look at the green bars in Sinovac, those green bars are viral replication in the lung of monkeys that haven't received vaccine, so the monkeys that receive placebo. What you don't see are the little orange bars, which indicate viral replication in the lungs of monkeys that have been vaccinated with the whole inactivated virus. So the whole inactivated virus protects against viral replication in the lung. With a ChAdOx vaccine, which is only a single dose vaccine, so it's given as a single dose, you do see some viral replication in the lung. Now, does that mean that there's going to be a big difference clinically? We don't know. In fact, we're really going to have to wait until we actually see the data in humans before we'll be able to tell you that one vaccine is associated with some limited viral replication, or that another vaccine, although it prevents disease, doesn't prevent viral replication. Now, why is that important? Well, a vaccine that doesn't prevent viral replication could mean that people who are vaccinated and exposed could continue to expose other people to the risk of infection. And we hope that that's not the case, but again, we'll only know that when we look in humans. Some of the companies are beginning to report COVID-19 data from humans. So I'm showing the Moderna data because everyone's very interested in it. What you see in the panels are different kinds of antibody. So in the upper left-hand panel is binding antibody. You see some neutralizing antibody titers, as well as pseudovirus neutralization. So the Moderna vaccine, as was reported in the press release, is associated with the production of neutralizing antibody with some minor with side effects. So almost everyone, actually 100% of the volunteers, experienced mild to moderate side effects. All of them were transient. And again, the company got rid of the highest dose, 250 micrograms, because actually one of the volunteers in that group developed grade 3 fever. So they're happy being at 100 micrograms with mild to moderate symptoms, but very good neutralizing antibody responses. CanSino has an adenovirus type 5 vector RBD. It's a Chinese private company. The issue here is that CanSino, at its highest dose, had 75% of recipients having neutralizing antibodies, which not quite what Moderna has, but okay. 75 is not bad. The issue is that 42% of the people in the highest dose group of CanSino had fever, and 14% had grade 3 fever, which is fever greater than 38.3 degrees centigrade. A significant side effect. If you back down the dose, you didn't see as many side effects, but you had much lower levels of neutralizing antibodies. So there's a trade-off there. So CanSino and Moderna are both moving on into phase 3. This shows data from the human trial of the Oxford ChAdOx vaccine. And again, you're seeing antibody levels, neutralizing antibody levels at the same level that you saw them in the monkeys, which is really great. A significant number of people, 16%, had fever. Almost 100% of people generated the correct neutralizing antibody, which is, again, something that you want to see. Systemic side effects were common, but they were mild to moderate and transient. So again, what you want to see with a vaccine. Unfortunately, a lot of the other companies have published their human data not in journals or not in preprints, but in press releases. So this is Sinovac's press release. This is Pfizer-BioNTech's press release. So Sinovac, of course, has a whole inactivated virus vaccine. This reports their data in humans. It says, we make neutralizing antibody, the vaccine was safe, but not in any form that we can actually review the data in depth. Pfizer did the same thing. The BioNTech vaccine, again, RNA vaccine was immunogenic. It was associated with some significant side effects, and it caused the company to to choose the lower of 3 doses. So again, important information, but not published in a publication, but in a press release. So that's about the vaccines. Now, in order to really get a vaccine, you have to first prove that it works, you have to then make it, and then you have to develop use. So this will be focusing on COVID-19 vaccine manufacturing. The Coalition for Epidemic Preparedness Innovations has identified 10 billion doses of vaccine manufacturing capacity worldwide. Now, that cannot all be used for COVID-19 vaccine manufacturing, and not all that manufacturing capability is specific for the vaccines that CEPI needs to have manufactured. You know, could they make the Oxford vaccine as well as the Sinovac whole inactivated vaccine? We don't know. But there are 10 billion doses of manufacturing capacity worldwide. COVAX, which is a consortium of CEPI, Gavi, and the World Health Organization, intends to have 2 billion doses of vaccine available by the end of 2021. This would allow the 150 or so countries that have signed up for COVAX to have roughly 20% of the vaccine they need for their populations. This would allow them to vaccinate those people at highest risk, the elderly and healthcare workers, by the end of 2021. Operation Warp Speed, which is a U.S. program, again, is putting billions of dollars into making vaccines at risk, making sure that at the end of the day, if the Moderna vaccine, for instance, is shown to be safe and effective at preventing COVID-19 infection, the U.S. will be able to say, we already have 100 to 200 million doses available for use in high-risk populations. Finally, a lot of the different companies are licensing out or contracting manufacturing. So, for instance, the Serum Institute of India is making the Chadox vaccine from AstraZeneca. Butantan is making the Sinovac whole inactivated vaccine, and SK Bioscience is also making the Chadox vaccine here in Korea, and Lonza, which is a European company, is making the RNA vaccine for Moderna. So companies are trying to increase their capacity to manufacture vaccines. Will it work? Will it be enough? At this point, we don't know. Finally, around the issues of access and equity, there's COVAX. COVAX is, again, a consortium of WHO, CEPI, and Gavi. More than 150 countries have joined COVAX, which is important because COVAX hopes to have 2 billion doses of WHO pre-qualified vaccine available by the end of 2021, roughly 20% of need. So COVAX aims to provide global access to all countries, to be impact-oriented and transparent around prioritization and around a need for the vaccine in different populations, and finally, to provide solidarity and collective ownership of vaccines which are really a global public good. We are contrasting that with the vaccine nationalism that some countries are showing these days. Finally, important point that vaccines don't save lives, vaccination saves lives. So how are we going to vaccinate 8 billion people, everyone in this world? First, we have to prioritize. We have to ensure that the logistics for shipping vaccine from the sites of manufacture to the sites of use are available. Actually, what's shown in the middle picture there is a man delivering vaccines on a donkey. Always to remember that the last mile, getting the vaccine from that vaccine refrigerator in a big city out to the countryside where it's needed, is a critical part of making sure that people get vaccines that are going to work when they're received. So delivery is very important. And then the unknowns. You know, we talk about vaccinating 8 billion people. Well, do we need a single dose for each of those 8 billion people, or do we need 2 doses? Do we need 8 billion doses of vaccine or 16 billion doses of vaccine? And will the vaccine work for a year, 2 years, 3 years? Or do we have to give a booster? So do we need another 8 billion doses to vaccinate everybody in the world again? Again, we don't know that, and we won't know that until we know what vaccines work and are safe. What you see there in the top picture are people lined up to receive the polio vaccine. Again, it may be like that for COVID-19. Vaccine hesitancy, a big issue, one that we have to deal with. We have to make sure that the vaccines are safe and effective, and we have to make sure that people are convinced, scientists are convinced, and that we can convince everyone that the vaccines that we are going to use to prevent COVID-19 are safe and effective. Safety, again, very important. We're going to need to make sure that countries around the world have vaccine adverse events reporting systems that are robust, so that if the vaccine looks like it's worsening disease or that it's associated with some very rare side effect, that we will be able to pick it up, analyze it, and try to prevent it in the future. Finally, minding the gap. And this is why everyone's so concerned about access and equity. because we've had a rotavirus vaccine since 2007, since it was approved by the U.S. Food and Drug Administration. By 2009, when WHO approved the vaccine for use worldwide, 70% of American children received the rotavirus vaccine. We are still not at that level for the rest of the world. In fact, worldwide, less than 40% of children have been fully vaccinated for rotavirus. We don't want there to be that large a gap between the haves and the have-nots for COVID-19 vaccine for 13 years after the COVID-19 vaccine is shown to be safe and effective. So we need to focus on access and equity and vaccination of everyone who needs the vaccine. So thoughts on the future. First, can we prove that the vaccine works and is safe? Yes, I think so. But can we do it in 12 to 18 months? Well, again, cautiously optimistic that if everything works as planned, we should be able to tell you the vaccine works and is safe. But does that mean that you'll have the vaccine? No, because can we make a vaccine in sufficient quantity, with high quality and an affordable cost? So we can't say yes to that. CEPI has identified global manufacturing capacity of 10 billion doses. COVAX intends to have 2 billion doses available by the end of 2021 for use around the world, again, for countries, for the 150 or so countries that signed up for the COVAX facility. So maybe. And finally, can we use the vaccine with respect to access and equity? Again, we're hoping that the COVAX facility and that other mechanisms that countries are beginning to explore will allow there to be access to this vaccine for the people in the world who need it. And right now, the target population for this vaccine is everyone, all 8 billion of us. So if you like baseball or if you're a physicist, they may have made the same statement, Niels Bohr or Yogi Berra. It's tough to make predictions, especially about the future, so any of these could be wrong. I hope not. Thank you.
Thank you very much for your presentation, Mr. Dr. Jerome Kim. The situation is very urgent and the vaccine development status has been covered by Dr. Jerome Kim.